when it comes to cancer therapies, we're always looking for those big breakthroughs, those aha moments.
Absolutely.
Right. And one of the most exciting areas is how we might take therapies that are already incredibly powerful, like CAR T cells,
Yeah.
and not only make them even more effective at fighting cancer,
Right.
but also safer in the long run,
Absolutely.
especially when we're thinking about how we can make them even more precise and targeted.
Precisely.
So you sent over some truly fascinating material
I did.
about how we might be able to do just that with these CAR T cells.
Yes.
And, CAR T cell therapy itself has shown remarkable success
It has.
in treating certain blood cancers,
Absolutely.
leading to significant remissions.
Yes.
However, we still have these challenges of relapses.
We do.
And particularly when it comes to tackling solid tumors,
Right.
that's still a major hurdle.
Absolutely. A major area of research.
Right.
Still a lot of work to be done.
Yeah, so we're talking about re-engineering, essentially,
Yeah.
a patient's own immune cells
Yes.
to become
Yeah.
precision-guided missiles against cancer.
Absolutely.
It's a really revolutionary concept.
It is.
And this research that you shared dives into
Yeah.
an exciting new area,
Yes.
how tweaking the epigenome, that layer of instructions on top of our DNA,
Right.
could be the key to boosting CAR T cell performance.
Yeah. And, thinking about it in terms of like software and hardware.
Yes.
Right. Where the hardware is our DNA.
Okay.
The epigenome is kind of like the software, the instructions that tell the DNA what to do, how to operate.
I like that analogy.
Yeah.
So this study zeroes in on a specific epigenetic regulator called TET2.
It does.
So for our listeners out there, what makes TET2 so pivotal?
Sure. So TET2 is actually really important for maintaining proper gene regulation in lots of different cell types.
Mm-hmm.
And what's interesting is that it seems to be involved in how T cells form memory.
Okay.
And that's relevant here because if we could make CAR T cells more memory-like, they could potentially stick around longer and keep fighting cancer longer, right?
That makes sense, yeah.
Longer-lasting anti-tumor responses. So the initial thinking was, well, maybe if we disrupt TET2 in these engineered CAR T cells, maybe we could create more persistent and effective cancer fighters, kind of boost their memory.
Yeah.
But as the study reveals, it's a bit more complicated than that.
Okay, so walk us through that.
Sure.
So the researchers tested this idea in models of leukemia and prostate cancer,
They did.
using different designs of these CAR T cells.
Yeah.
So what did they discover?
So first, they wanted to see how well these TE2 disrupted CAR T cells actually fought the cancer initially.
Okay, so initial response.
Yeah, right out of the gate.
Yeah.
And they actually found some really intriguing results.
Okay.
And importantly, it really depended on the specific CAR design they were using.
Okay.
So in one experiment, they looked at CAR T cells that were designed to target CD19.
Okay.
Which is a marker found on leukemia cells. And specifically, they were looking at CARs that used a signaling component called 4-1BB.
Right.
Now, they called this particular CAR RV19Bs.
RV19Bs.
Yeah.
Okay.
And when they disrupted the TT2 gene in these RV19Bs' CAR T cells, they actually saw that these modified cells led to better survival in mice with leukemia compared to the standard unmodified CAR T cells.
So they lived longer.
Yeah, they had better outcomes.
Okay, so promising initial finding.
Absolutely.
For that specific CAR design.
Right, and that's an important point.
Right.
We're seeing already that the CAR design itself really matters. It's not a one-size-fits-all situation.
Got it.
Now, another initial design they tested used a different signaling component called CD28.
Okay.
This one was called RV1928.
1928, okay.
And with this one, when they disrupted TT2, they didn't really see a significant difference in survival between the TT2 edited and the unedited groups, at least not in those early stages.
Right.
But the researchers, being the thorough scientists that they are,
they didn't just stop there.
Uh-huh.
They thought, okay, well, maybe the way the GRIAR gene is expressed within the T cell, maybe that matters too.
Okay, yeah.
So they looked at some versions of the 1928 CLUVS CAR that were engineered to potentially be more persistent.
Okay.
So either by having them also produce another signaling molecule called 4-1BBL.
Right.
So that one's called RV1928s plus 4-1BBL.
Okay.
Or by inserting the CAR gene into a very specific location in the T cell's DNA.
Okay.
Called the Tourette locus.
Right.
Which is known for stable, long-lasting expression.
Okay.
So that one is Tourette 1928 says.
Okay, so these are versions of that 1928's CAR that are potentially just going to hang around longer.
Exactly. Potentially stick around and keep doing their job longer.
Okay, so what happened when they disrupted TT2 in those more persistent versions?
Well, in those cases, disrupting TET2 actually did enhance their ability to fight the cancer compared to when T2 was still functional.
Interesting.
So again, it seems like that internal signaling within the CAR T cell
really plays a role in whether losing TT2 actually gives you that initial therapeutic advantage.
Yeah, and that makes sense, right?
Right.
Because if the signal is already super strong from something like 4-1BB, or if the production of the CAR is really stable and long-lasting,
right,
maybe it's already doing its job as well as it can.
Exactly.
Yeah.
And disrupting TT2 might not make a huge difference at that point.
Okay, so it's all about fine-tuning these signals and making sure that they're working in concert.
Yeah, and that losing TT2 is actually beneficial in that context.
Right. Okay, so it sounds like tweaking the epigenome by disrupting TT2 could be a way to boost these CAR T cells.
Potentially.
At least initially.
Right.
And depending on the design of the therapy.
Exactly. That seems to be the recurring theme here.
Yeah.
The CAR design really matters.
Okay, but you mentioned something really interesting.
I did?
About unchecked proliferative responses.
Right.
So this is where it gets really interesting.
Yeah. This is where things take a bit of a turn.
Okay, tell me more.
So as encouraging as those initial results were in terms of the cancer-fighting ability, the researchers kept monitoring these mice over a longer period.
Okay.
And that's when they started noticing some concerning developments.
Okay.
Even after the tumors had seemingly been eliminated.
Oh, so even after the cancer was gone.
Right, exactly. So some of these mice that were treated with those TET2 edited T cells, they started showing symptoms like enlarged spleens and livers.
Okay.
And when they looked closer, they found that these organs were just packed with CAR T cells, even though the original cancer was gone.
So they were multiplying even without the cancer there to stimulate them.
Right. So it's what we call antigen-independent expansion.
So these CAR T cells were multiplying even though their target was gone.
Exactly.
And that sounds like it's the opposite of what you want.
Yeah, you want them to be controlled. You want them to respond to the threat and then back off when the threat's gone.
Yeah, you don't want them just going rogue.
Exactly, but that's what was happening.
Okay.
These CAR T cells were undergoing clonal expansion.
Right.
Meaning specific cells were rapidly dividing and creating large populations.
Right.
But this multiplication was no longer dependent on the tumor. So they saw this big surge in the number of CAR T cells in the bone marrow and the spleen in mice treated with TE2 edited RV19 BIZs.
Right.
RV1928 says plus 41BBL.
Okay.
And TRE1928's CAR T cells.
Got it.
Now, again, they didn't consistently see this with that standard RV1928 says CAR.
Right. So again, the design matters.
The design matters, exactly. So it seems like there's something about those particular CAR designs that might make them more susceptible to this uncontrolled growth.
When T2 is out of the picture.
Exactly.
Okay, so they're multiplying like crazy, even though the cancer is gone. So what did the researchers discover about what was actually fueling this unchecked growth?
Okay, so they dug deeper into the molecular characteristics of these rapidly multiplying CAR T cells.
Okay.
And one of their key findings was that this uncontrolled growth actually required the loss of both copies of the TET2 gene.
Okay, so 2 copies.
Yeah, within the T cell.
Okay.
So if only one copy was disrupted, this problematic proliferation didn't really persist.
So both versions of the gene had to be knocked out.
Yeah, we call that biallelic disruption.
Okay.
So that was a crucial detail.
Right.
Now, they also found that a protein called BATF3 was playing a vital role in this whole process.
Okay.
So these hyperproliferative cells, they showed sustained high levels of BATF3. And just as a reminder, BATF3 is a type of transcription factor, which means it controls which genes are switched on or off.
Right, so like a master switch.
Exactly. And it turned out that BATF3 was driving a growth program that relied on another very well-known gene involved in cell growth called MYC.
Yeah.
Yeah, so you can kind of see the chain of events here.
Okay.
Loss of TE2 leads to this sustained activity of BATF3, which then boosts MYC. And all of that leads to this runaway cell growth.
And these immune cells that are supposed to be helping you.
Right. It's like they've lost their brakes and they're just multiplying out of control.
Okay, so quantity over quality.
Right.
They're multiplying a lot.
Yeah.
But are they still effective at fighting cancer?
That's the big question, right? And unfortunately, the answer is not really.
Okay.
So despite their large numbers, these hyperproliferative CAR T cells actually showed reduced effectiveness in killing cancer cells. They also released fewer of those important immune signaling molecules that are crucial for a strong and lasting anti-tumor response.
So they weren't doing their job as well.
Right. They were less effective at actually eliminating the cancer cells. And the study mentions that this state is different from typical T-cell memory or exhaustion.
Yeah, it makes it distinct.
So they were looking to see, you know, are these cells just kind of like memory cells gone wrong? Or are they exhausted T cells?
Yeah.
But they didn't really fit either of those categories.
Okay.
So they looked for specific markers and gene activity patterns that are usually associated with memory T cells, which are the ones that can remember and respond to previous threats. And also exhaustion, which is when T cells become less effective after prolonged stimulation. And while these hyperproliferative cells did show increased levels of some memory-associated markers, they didn't express some of the key memory-related proteins, and their overall gene activity profile didn't match either memory or exhaustion.
Okay, so they were something different.
Yeah, they were in this kind of unique state. In fact, their gene expression patterns actually showed some similarities to certain types of T-cell lymphomas.
Okay, now that's concerning.
Right. That's a red flag.
Yeah, given the potential for uncontrolled proliferation.
Exactly, because lymphoma is a type of cancer that affects the lymphatic system. And it's characterized by this uncontrolled growth of cells. So seeing similarities to lymphoma-like gene activity and these hyperproliferative CAR T cells definitely raises concerns about potential long-term risks.
Yeah, so did they actually find evidence of these cells becoming cancerous themselves?
So they didn't find any recurring cancer-causing mutations.
Okay.
But they did observe that these cells were more prone to acquiring secondary genetic mutations.
Interesting.
So it's like losing TET2 not only makes them multiply uncontrollably, but it also makes them a little bit sloppier when it comes to maintaining their genetic material.
Okay, so they're more likely to make mistakes.
Yeah, exactly.
When they're copying their DNA. Okay, so that's another red flag.
Yeah.
In terms of long-term safety.
Right, because those mutations could potentially lead to them becoming cancerous.
Yeah, okay, so it's like TET2 is acting as this guardian.
Yeah, that's a good way to put it.
Right. Not only keeping them from multiplying out of control, but also helping keep their genetic material stable.
Yeah, so it's got this dual role.
Right. And this study also looked at how the specific CAR design influenced this hyperproliferation.
It did.
And, you know, as we were talking about before, it seems like the CAR design is really crucial here.
It's so important.
Why is that?
Well, as we've been discussing, the RV1928's SS seemed less likely to trigger this hyperproliferative state compared to the others. Like the RV19B's, the RV1928's plus 41BBL, and the TERC1928's.
Okay.
And they even did some really cool experiments where they introduced different CARs into the same batch of TET2 edited T cells and tracked individual T cell clones. And they found that the clones that were expressing those CARs that were linked to hyperproliferation were much more likely to expand and persist long-term compared to those expressing that standard RV1928 size CAR.
So it's really pointing to the signaling pathways initiated by the different CAR designs as being really important for whether those TET2 deficient cells
are going to go into that uncontrolled growth state.
Yeah. And it makes you wonder, right, if the signals that tell the T cell to activate and persist, if those signals are really strong from components like 41BB or 41BBL, or if that CAR production is really stable and long-lasting, like from that TREC locus, makes you wonder if maybe that creates an environment where the loss of TET2 is more likely to tip the scales.
Towards that uncontrolled growth.
Exactly.
Okay, so let's zoom in even further, Yeah. At the molecular level, what's the connection between losing TET2 and this increased activity of BATF3 and MYC? Right. So how does that actually happen?
Yeah.
Well, the researchers did some really interesting experiments to examine how accessible the DNA was in these hyperproliferative cells. And what they discovered was that the regions of the genome that contain the BATF3 and MYC genes, including those regulatory elements that control their activity, were much more open and accessible in those TET2 deficient CAR T cells compared to the normal CAR T cells.
But it's like TET2 is normally keeping those regions closed off.
Yeah, it's like it's putting up a fence.
Yeah.
And when T2 is gone, that fence comes down, and all of a sudden those genes are much more easily accessed and transcribed.
Okay, so that means more BATF3 and MYC can be produced.
Right, exactly.
Okay, so that explains how that happens.
Oh, and they even identified specific DNA sequences called AP1 family binding motifs that BATF3 actually binds to, and these motifs were much more prevalent in these open regions of the DNA.
Okay, so it's like BATF3 has more places to latch on.
Yeah, exactly.
And do its thing.
Right.
Okay, so it's like the self-perpetuating cycle.
Yeah.
Right?
It is.
TET2 is gone. BATF3 and MYC can be turned on more easily.
Right.
And because BATF3 drives MYC, you get this runaway proliferation.
Yeah. And it's no longer tied to fighting the cancer.
It's just doing its own thing.
Exactly.
So that's obviously a problem.
It is.
But the study also looked at ways to stop this. Right.
Did.
So what did they find?
So given how central BATF3 seemed to be, they wanted to see if blocking its activity could actually halt the hyperproliferation. So they used both genetic methods and drugs.
Okay.
So genetically, they showed that if they disrupted BATF3, in addition to TET2, that hyperproliferative behavior was significantly reduced.
Okay, so that makes sense, right?
Yeah, you're taking away that drive.
Right, if BTF3 is that master switch, and you take it out, then the whole process should at least slow down.
Exactly.
Okay, and what about the drugs?
So they tested a compound called JQ1.
JQ1, okay.
Which inhibits a protein that's really important for producing both BATF3 and MYC. And they found that JQ1 effectively inhibited the uncontrolled proliferation of these TE2 deficient CAR T cells and reduced the levels of both BATF3 and MYC.
So it's kind of attacking that pathway from a different angle.
Right, exactly.
Instead of just knocking out the genes, you're using a drug to interfere with the proteins themselves.
Right, and they also looked at dexamethasone.
Dexamethasone.
Yeah, which is a common anti-inflammatory corticosteroid.
Right.
And interestingly, while dexamethasone didn't really affect the initial growth of the TT2 edited CAR T cells, it significantly inhibited the growth of those hyperproliferative cells, the ones that were TT2 deficient.
So it was specifically targeting that unchecked growth.
Yeah, it seemed to be more effective at controlling that.
Okay, so that's interesting.
And again, they saw that this was linked to a decrease in BTF3 and MYC levels.
Okay, so it seems like there are a few different ways that we might be able to control this.
Yeah, and the sensitivity to dexamethasone might actually explain a case that was reported previously.
Oh, tell me about it.
Yeah, so there was a patient who had a TET2 deficient CAR T cell clone. And that clone actually decreased in size after the patient received corticosteroids to manage a side effect called cytokine release syndrome, which is something that can happen with KCO T cell therapy. So it's possible that the corticosteroids, which include dexamethasone, might have actually played a role in controlling that TET2 deficient clone.
Interesting.
So it kind of fits with what they were seeing in this study.
Okay, so just to recap, we've got this situation where disrupting TT2 can make these CAR T cells more effective initially.
Especially for certain CAR designs.
Yeah, right. But it also carries this risk.
It does.
Long-term antigen-independent hyperproliferation.
Right, meaning they keep growing even when they're not supposed to, even when the cancer's gone.
Right, and that's driven by BATF3 and MYC, which could lead to reduced anti-tumor function. And even genetic instability.
Right, more mutations.
Right, but targeting BATF3 or using drugs like JQ1 and dexamethasone might offer ways to manage that side effect.
Absolutely.
Okay, so what are the broader implications of these findings for the future of CAR T cell therapy?
Well, I think this study really emphasizes that as we explore using epigenetic modifications to improve CAR T cells, we have to be extremely careful about potential long-term effects.
All right.
So one strategy that might be helpful is instead of completely knocking out TT2, maybe we could just inhibit it temporarily during the manufacturing process of the CAR T cells. So they get that initial boost, but they don't have that sustained risk.
Of uncontrolled proliferation.
So kind of hit it and quit it.
Yeah, exactly. And another thing is, we might need to start screening patients for any pre-existing genetic factors that might make them more susceptible to this type of uncontrolled growth.
Okay, so that we can tailor the therapy accordingly.
Exactly.
And of course, the fact that the core design itself plays such a crucial role means that we need to be very careful about how we engineer these receptors.
Absolutely.
So it's a lot of work to do.
It is, but it's exciting work.
It is. It's really cutting-edge stuff.
Yeah, we're talking about potentially revolutionizing cancer treatment.
Right. So knowing that even beneficial genetic tweaks can have these unintended consequences.
Yeah.
In powerful therapies like CAR T cells. How do we balance innovation with vigilance to ensure that we're developing the safest and most effective treatments possible?
That's the million-dollar question. It's a constant balancing act.
Yeah, and I think the study really highlights that
it does.
We've got to be thinking long-term.
Right.
We can't just focus on those initial results. We've got to make sure that these therapies are safe and effective
in the long run
for years to come.
And thinking about how the CAR design itself plays a role.
Yeah.
What other novel engineering approaches might be out there?
Right. How can we tweak the design to minimize the risk of uncontrolled proliferation while still maximizing that anti-tumor power?
Right.
It's going to be really interesting to see where this research goes.
Absolutely. It's a fascinating field, and this study gives us a lot to think about.
It does.
As we move forward.
Absolutely.
All right. Well, thank you so much for joining us today.
It was my pleasure. Thanks for having me.
It's been a really insightful conversation.
I agree.
And for those of you listening, we hope this deep dive has given you a better understanding of the complexities of CAR T cell therapy.
And the potential of epigenetic modifications.
Right. And the importance of balancing innovation with safety as we develop these groundbreaking treatments.
Absolutely.
All right. Until next time, stay curious.
And stay informed.
We'll see you on the next deep dive.
That's it.